So we have already talked about how cells progress through the cell cycle once they receive a mitogenic signal. They begin by going through the G1 phase and then ultimately replicate their DNA in S phase, and then undergo further doubling of their organelles and cellular components in G2 before entering mitosis when the chromosomes and cytoplasm are divided and one cell can become two. So this whole process of the cell cycle is highly regulated. Because you can imagine that if any part of this process was done incorrectly, it could lead to uncontrolled cell division or division of abnormal cells, both of which are hallmarks of cancer. And so in order to make sure that cancer does not happen, the cell cycle has to be regulated at many points. We talked a little bit about the G1 to S checkpoint in the last several lectures ago, and that is marked by P53. This is sort of that start checkpoint. So in addition to checking if the DNA is damaged and using P53, the cell will also sense if the environment is favorable for division in the first place. And so this is often seen as more of an external checkpoint as well. Should I even bother dividing? Is there a growth factor mitogenic signal there triggering me to divide? And then, before I finally enter into S phase, is my DNA damaged? And if the cell meets all of those checkpoints, yes, the environment is favorable, yes, there's a growth factor, mitogen, no, your DNA is not damaged, it can progress from G1 into S. And then there's another checkpoint at the end of G2 to sort of ensure that before you enter into mitosis, where the real division takes place, everything has been replicated effectively. You have enough organelles, molecules, cellular components, and an adequately replicated DNA to create two new cells. The environment still needs to be favorable, but if these checkpoints are made and the DNA has been replicated effectively in S phase, and all of the components that are necessary are made in G1 and G2, the cell can move past this checkpoint as well and enter into mitosis. And we talked a lot about the metaphase to anaphase transition in mitosis as being really important checkpoint in the cell cycle as well, making sure that the chromosomes are attached to the spindle, and not only that, but that they're lined up perfectly in the center so that the DNA or that genetic information splits evenly between the two new cells. And so in addition to these checkpoints or these sort of molecular breaks that exist within the cell cycle, there are two different proteins that actually serve to regulate this process and promote all of the processes that happen in each of these phases when they are active. And those two proteins are called cyclins and cyclin-dependent kinases. And a cyclin-dependent kinase is an enzyme. It's a kinase, right? So it phosphorylates things. And what it does is it generally phosphorylates things to activate a certain step of the cell cycle. And it only does that in the presence of a cyclin. So CDKs exist in an inactive form within your cells at all times during the cell cycle. And then when their appropriate cyclin is generated, that cyclin binds to an inactive CDK, activating it so that it can promote movement to that phase of the cell cycle. And so you can see here that there are three main cyclin-dependent kinases, and there are three separate cyclins that regulate each of them or activate each of them. So there's the G1S cyclin-dependent kinase and a G1S cyclin. There's an S CDK and an S cyclin and an M or M phase CDK and an M cyclin. And what you'll notice is that the levels of cyclins change as the cell cycle progresses. And so the levels of cyclin molecules can be seen here. G1S cyclin peaks towards the end of the middle of G1 and then goes down right before S phase. S cyclin increases and then peaks and stays very high within the cell all the way through S, G2, and most of M phase. And M cyclin comes up in level through G2 and then ultimately decreases right after M phase. And what's important to note is that the levels of CDKs do not really change at all within the cell cycle. those CDKs are present at the same levels, the same rate throughout the cell cycle, but they're inactive. It's the cyclins that change their expression level, and then by doing that, activate the CDKs when they're needed. And then once the cyclins are degraded, or their levels go down during the cell cycle, that CDK becomes inactive again. And so cyclins serve to regulate the CDKs, and then subsequently the CDKs regulate which phase of the cell cycle the cell is in. And it's much easier for a cell to regulate cyclins than it is to, or levels of cyclins than it is to regulate the levels of a CDK. CDKs are enzymes, right? And so they have to have a particular structure in order to function. And making a more complicated protein like a kinase is going to take more time in terms of protein synthesis than it is to make a small molecule like a cyclin. And so by keeping the CDKs around in an inactive form and just making and degrading cyclins throughout the cell cycle, it allows the regulation to happen a lot faster and quicker than if you had to wait for the cell to make and then degrade CDKs. You can see here that in G1 phase, there's a mitotic signal present that promotes expression of cyclin and then activates this SCDK. And then at the end of G1, as the cell is transitioning to the next phase, ultimately there are proteases that turn on and can degrade cyclin, or this S-cyclin, to turn at the end of S phase to promote movement of the cell into the next several phases. And once again, I just want to remind you how imperative it is that the cell cycle be regulated by the molecular breaks and checkpoints, as well as by cyclins and CDKs, and the APCC complex, and P53. Because unregulated cell cycle is what leads to cancer, right? And what I'd also like to tie in here is that cyclin synthesis, as well as CDK activation, kind of as a result, is regulated not only by the cell cycle, but also by different external factors. So in order for G1 CDK to be activated and these cyclins to be synthesized, the extracellular environment has to promote cyclin synthesis. In order to move from G1 to S, the DNA damage checkpoints need to be checked off. There needs to be no DNA damage in the cell, because you can see here, if there is DNA damage, it inhibits both the CDK, G1S CDK, and the S CDK, and stops the cell cycle before it reaches S. If the DNA damage can be repaired, that inhibition can be relieved and the cell can move through the cell cycle. In the case of regulating the cell cycle at the level of mitosis, any unregulated DNA, replicated DNA at the end of G2, then that checkpoint will inhibit mCDK, which would promote mitosis. Any DNA damage sensed here will also inhibit mCDK and stop the cell cycle progression right here. And additionally, if a chromosome isn't attached to the spindle, if the cell can make it into mitosis, that will inhibit APCC and stop mitosis mid-mitosis in metaphase and never allow anaphase to be promoted. And so there are all of these important mechanisms to sense DNA damage, unreplicated DNA, and the potential to split your DNA incorrectly by not having all chromosomes be attached to the spindle, that feed back on the CDKs and basically inhibit them from promoting this progression through the cell cycle until the DNA damage is either fixed or until the cell can trigger apoptosis and basically commit suicide, because the cell would rather undergo programmed cell death than continue to replicate with damaged DNA or unreplicated DNA or an improper separation of chromosomes And so that regulation of the cell cycle is once again really imperative to having proper division of healthy cells from one cell to two identical daughters.